So today is going to be our second lecture on genetic variants and genomic variation for the Bioinformatics class at CSUMB. And this will be part two of this topic, and hopefully we'll finish up our discussion talking a lot about genetic variation, how it directly relates to how we think about things at a genome scale. Some learning outcomes that we're going to emphasize here. Again, we're going to be emphasizing how we use genomic variants in biological research. Also how we are proceeding from using genetic data to understand phenotypic outcomes, as well as understanding how specifically when we think about genomics, we're using Genome-Wide Association Studies and how we might interpret them, as well as some of their limitations. So when we think about the application of genomics to biology, one of the main contributions that we really see genomics having a big role in is understanding this relationship between genotype and phenotype. So how are genetics represented in the traits that we observe in organisms? So how do we get from the genetic information that we see in a phenotype? And there's a couple different ways. So historically, before kind of like the genomics era, we were using methodologies like QTL mapping, which helped. It's a process. QTL mapping. QTL stands for quantitative trait loci, and this was a process by which we would identify genomic regions or quantitative or loci that potentially contain genes responsible for phenotypes, so that could quantitatively contribute to a trait. And it was built on this kind of classic equation that we pull from quantitative genetics, which is that when we think about the variation that we see in phenotypes, it can be distilled down to a bunch of different types of factors. So there can be genetic components that are contributing to phenotypic variation. There can be environmental components that are contributing to phenotypic variation. There can be interactions between the two. There can be experimental error. And there can be actually a host of other ways that we can partition out the variation in this case. And QTLs, what they actually represent, these regions represented, were sort of these polygenes that could be affecting complex traits. One of the things that QTL mapping experiments try to do is they try to control for genetic variation, decrease environmental variation. And by doing these things, we're better able to increase and explain the proportion of phenotypic variance that's attributable to genetic variance. And specifically, the goal here is to increase the number of gametes with additive genetic trait values. This is generally accomplished by breaking the gene linkages via recombination for each generation. So we're really kind of breaking apart gene groups until we can identify specific regions that can then be associated, that have additive genetic values that are associated with phenotypic traits. There's a couple different designs. There's the F2 design, there's the backcross design, but typically we would always want to start off with some recombinant inbred lines. An example of what one of these experiments might look like is shown over here on the right, where we start off with two recombinant inbred lines, two parents where the chromosomes are basically the same. We then cross those parents and we end up with our F1s, and then we continue to do crosses of those F1s with each other, crossing the next generation with each other, crossing the next generation with each other, until we end up with individuals that have mosaics. And this is due to recombination occurring over time, right? So they're mixed back and forth. And basically what we can do is after a few generations, we can start to look for the association between specific regions of these chromosomes with specific phenotypes. And by identifying that there's always a specific region associated with a specific phenotype, this example of just a few hairs on the back of these leaves, helps us to identify what the quantitative trait loci are. This is how we're able to identify the regions of the genome that are contributing to this trait. One of the ways that we start associating quantitative trait loci with genetic variants is that we can also use SNPs to map along those chromosomes to identify where these loci might be located, right? And one of the great things about single nucleotide polymorphisms is that we can look at lots of them at one time, and so we can really build a map of the genetic variation that's in an organism. And this helps us find an association between a marker and a phenotype. And that's sort of the statistical representation of this relationship, where we have this, this blue area up here, right, that looks to be associated with a very specific phenotypic outcome. And when we look, generally when we are using these SNP approaches, we're trying to identify, do we see specific regions associated with that specific outcome? So a lot of times what we're looking for is a significant effect of an allele on the phenotypic mean. And this is just like a classic t-test, right? You're just comparing two groups. Does this allele change the value of the mean in this case or without it? To do that, we can start to use more complicated approaches, specifically an approach where we would look at something referred to as the logarithm of odd scores, or the LOD. And this is an example of what the LOD equation is, right? And the log of odd score here, we can see the log comes from where we're taking a log base 10. We're of the probability of a sequence with a given linkage value divided of a sequence with no linkage, right? So that's kind of the relationship that we're looking at. And what this is really comparing is it's comparing the likelihood of obtaining the test data if the two loci are indeed linked versus the chance that they just appeared together purely by chance. So what's the probability that we're looking at two loci that are linked versus over divided by the probability that they are random? And typically what we think of this is if we get a score greater than 3, that's considered to be evidence for linkages. This indicates 1,000 to 1 odds, so 10 to the third, that the linkage being observed did not occur by chance. And this is an example of how we can see there's the log likelihood ratio statistic over here on the left, over here on the y-axis, right? This is along the chromosome as we look, and here we can find some highly significant and some significant markers that are suggestive or highly indicative of a specific region being associated with some specific trait or outcome. Association mapping is another approach that sort of builds on the quantitative trait loci approach. And here, what we're trying to do is find the contribution of common variants to specific diseases or phenotypes. Sometimes this is also called linkage disequilibrium mapping. In particular, one of the things that we're interested in is a particular SNP more commonly seen in affected, unrelated individuals than we would expect by chance. So that's the general hypothesis that we're trying to test. And we can look at this from a couple of different ways, from direct contribution, from indirect or linked contribution. And one of the ways why this is important to think about this indirect linked contribution is, yeah, it's great if we can observe the exact allele that we think is associated with a specific disease or trait or whatever it is we're looking at. But historically, we haven't actually sequenced the entire genome when we're generating SNP calls. Instead, we've used microarrays or other chip arrays to look for patterns of SNP variants that are tagged. And so I mentioned this a little bit earlier, but it takes about a million SNPs kind of spread across the human genome, across all the 22 autosomes and the sex chromosomes, to encompass all of the amount of linked regions in the genome. So in other words, you don't need to report all of the SNPs specifically to be able to say, hey, I can identify a region of the genome which is associated with a specific trait. So I actually don't need all of the SNPs, I just need one in that region to assess whether or not that region is associated with a specific outcome. This brought about what's commonly referred to as the case control population sampling approach in association mapping. And this is basically just saying, do we see an association between a specific SNP and a disease in a large population? There is another approach that we sometimes use in association mapping, which is referred to as transmission disequilibrium testing. And what this is looking at is whether or not we see unequal transmission of SNP alleles to affected and non-affected siblings. So this is doing kind of more of an identity by descent approach of thinking about where these alleles are ending up. This whole idea of association mapping and doing these types of examinations is largely based on what's referred to as the CDCV proposition. And the CDCV proposition is that common diseases are due to common variants of moderate or large effect. That's the proposition. So this is the case where association mapping has the most power. Alternatively, there could be other outcomes, right? So we could be looking at it as a case where there's lots of common alleles of small effect, or there's lots of rare alleles of large effect. So there are more than one possible outcome, but these are other possible outcomes that we could be examining. Here, one of the things that we're looking at is we're trying to find a SNP that is associated with a specific outcome, and then by linkage, we're saying that that SNP, the things are within 5 centimorgans of that SNP, are going to be that the disease allele is somewhere within those 5 centimorgans. The association approach is a little bit different when we do the association approach. The association approach indicates that there's a SNP that is significantly associated with a disease or trait, right? So it's not necessarily identifying a region, it's saying that this specific SNP is associated with it. So it kind of depends on whether or not we're using that tagged approach or whether we're not using that tagged approach. So there's a variety of factors that can affect our success in identifying associations between genetic variants and traits. One is how heritable a disease or trait is. And this especially has a role in sort of like the relative risk. So if we want to think about, what is the risk of someone, say like you get your 23andMe results and you're trying to figure out, well, how likely am I, if I have some sort of disease result, am I to be susceptible to this disease? And so the higher the heritability, the higher of the trait or disease, the higher the risk, right? And an example we might be able to use is something like mutations in BRCA1, often referred to as one of the main tumor suppressors that can be responsible for dysregulation in breast cancer and ovarian cancer. And people who have mutations in BRCA1 tend to have a relative risk of about 65%. So it means that they're at a much higher percent chance of getting breast cancer, but it's not 100% by any means. So that means that when they inherit these sorts of things, it's not 100% heritable they're getting, but they do have an elevated risk. So there's some other things. Polygenes can really have a big dampening factor on our ability to detect associations between traits and diseases, because we have to identify more than one thing or more than one region. So the more genes that are involved, the more difficult. Also, the penetrance and expressivity of the quantitative trait loci effects are really important. So you might have heard this too. We talk about penetrance and expressivity a lot in disease models, so you might have heard that recently with a lot of the COVID-19 stuff going on. But penetrance is basically a measure of the fraction of individuals with a quantitative trait that show quantitative trait locus that show the trait. whereas expressivity is the severity of the locus's effect. So how many individuals have the trait, or how strong is the trait shown. So penetrance, one of the things that we know is that it's sometimes difficult to measure because it can be strongly age dependent. So you may be more prone to a disease as, say, you get older. Schizophrenia might be an example of a disease like this, right? And expressivity tends to be more indicative of things that happen in the environment. So it can be highly modulated by the environment. So the extent and uniformity of linkage to disequilibrium between the SNP and the marker and disease is also really important. So variation or limited influence between that SNP, if it's a tag SNP, if it's not actually the actual variant, which is the causal variant, it can affect how successful we are in identifying these things. And one of the things that can really affect this is when these SNPs have variable strength of effects. Genetic or allelic heterogeneity can also be a factor affecting success. So here we could think about how there's about 100 different mutations in CTCFR gene and CFTR genes that can cause cystic fibrosis. So that means that there's a lot of different possibilities out there, and it may be difficult to identify the causal variants when there's lots of different things that cause the same phenotypic outcome in different individuals. Population stratification is also something that can mask associations. Here, differences in alleles and phenotype frequencies among populations can lead to a lot of false positives. So this happens all the time in association studies where we might be looking at one population and they say, oh, it looks like this gene is involved in autism or something like that, and then we go to another population and it looks very different. Admixture is also important to consider. Here we could have recent contact between previously isolated groups, and that's going to wash out a signal, or it could introduce some either transient linkage disequilibrium or a novel population structure that kind of hides the association between a specific variant and a specific phenotypic trait outcome. Not only does sort of spatial population structure, but any hidden environmental structure. And this can be really important for diseases where, highly determined by what environments we tend to find people in and that they've experienced. And so if we can't see that environmental structure or identify it, it's very difficult for us to partition out samples in an appropriate way to identify the comparison groups we're looking at. And then lastly, we'll say, the interactions between genetics and environment and the interactions between different genotypes within an individual are also really important. So the context matters. So how these genes interact with the other genes around them or with the environment that they're in has a big effect too on the strength of the relationship between the trait and the underlying genetics. And of course, if we don't completely genotype things, the more genotyping, the more power we have, the more resolution we have. So incomplete genotyping can really be a difficulty here. So if we think about how we've gone and identified some of the main candidate genes for a complex disease such as type 2 diabetes, here what we can see is when looking at a gene like NIDDM1, there's been a lot of things that have been done. So first we start off with linkage mapping, and we found about 7 centimorgans on chromosome 2 that look to be promising. This region looks to be promising where this gene is, but that's still a big section of that chromosome. Then we could do some things like construct a physical contig of that region, so we could sequence it and find there's about 22 genes there. We could do some case control tests and look, and here we can narrow it even further down. We can say, hey, it looks like 3 genes are associated with this disease, significantly associated with this disease. You could sequence that. Now we might have found these 3 genes, right? Then we can sequence individuals over this 100 kb area, so over 66 kb, and we can describe all the variants that are there, all the SNPs that are there, and then we can look at those specific SNPs and say, okay, now which one of those SNPs, not just which one of those genes, are associated with that outcome. Here we can find it's this SNP 43, right, and that is involved in this calpain 10 intron 3 position. Again, we can look for haplotype tests of complexity. We can try to find, which haplotypes are associated with this complex disease. We can do some tests by geographical populations. How frequently do we observe this disease in Northern Europeans and Mexicans, Mexican Americans? Do we see the same? Do we see differences? That might tell us about something about their general background, their genetic background, and how that might affect our outcomes. We can take some information from outside too that we might know from things like the gene ontology or the KEGG database, and we can say, well, we've been able to narrow it down to this NIDDM gene. Well, what is it actually? What could these SNPs in these regions be doing? And so one of the things that we can find is that, if the SNP is binding, is affecting where this nucleotide, nuclear factor can bind, or this transcriptional activation reporter, then that might have a functional effect. And so in the end, we come up with a hypothesis, right? So calpain 10 protease is somehow involved in NIDDM activity, and that shows us an association with this specific type of outcome. So what does an association study look like in terms of what the data looks like? And specifically, I think it's important here that when we're talking about association studies at a genome level, we're specifically talking about something called a Genome-Wide Association Study, or a GWAS. And what this does is that instead of sort of doing targeted associations, here we're examining as many common genetic variants in different individuals to see if any variant is associated with a specific trait. And so the genotypes are at least 1 million SNPs here, and they're spread throughout the genome, and we're looking for an association. So here we can see, if we look at this table down here, we can see, oh yeah, we've got our control in our case, and we can see, in this spot that there's some looks to be some definite associations of specific outcomes with the case versus the control. So the case control association study works again, kind of the traditional way of having like a control and treatment comparison. It's the most direct, most common. And again, the general question is a particular marker more or less frequent in individuals affected with a disease than in uninfected people. So you got to have a control group, you got to have a treatment group. And here we're looking for a specific SNP association with the disease trait. As I mentioned before, there are these pedigree-based analysis, these transmission disequilibrium tests. They leverage sort of individual, how independence from linkage disequilibrium can be measured, and specifically coming from this Mendelian perspective that are trying to isolate the effects of a single affected child from a family from the unaffected individuals in that family. And what you end up with is a probability of allele transmittance to children with the trait. So in the case of, the case control, what we're looking for, if we think about what the rationale here is like at some point there's a mutation which is going to lead to these cases, right? It's going to happen at some point. And it could have happened multiple times in sort of, say, different groups of individuals. And what we would see is that in individuals that have had this mutation, because it had happened in this lineage, we would find it more commonly among individuals that were related to each other versus versus groups that were not expressing that. So it's at a higher frequency. That's why we would associate with a specific case. And we'd be able to replicate that here in other cases as well. Because here we can see this mutation in both cases. These two individuals are demonstrating So from an epidemiological perspective, specifically a genetic one, when we think about GWAS studies, kind of our underlying model of understanding is one that's trying to look at each allele as a risk allele that's associated with one of these outcomes. And this risk allele conveys how much risk there is for the phenotype from an additive genetic perspective. So if there are a lot of times there can be minor alleles that have a recessive effect. Right? We talk about dominance and recessive genetics, and you got to have 2 copies in order to actually demonstrate that risk. So the dominant effect can be a different one, and here you only need 1 copy to be at risk. One of the big efforts one of the reasons people like this GWAS approach is that it was seen to be kind of a hypothesis-free approach, because you didn't have to know much about your genome ahead of time in order to look for these associations, right? So you could just say, hey, we can look genome-wide. We don't have to rely on prior knowledge. It's going to save us a lot of time. So it's going to be great. And as we saw, the cost of genome sequencings or genotyping go down. Notice that they're even way lower per sample than what we were talking about for how much it would cost to sequence a genome, and that's because you're only running these. A lot of times you're just running these chips, and these chips are super cheap. One of the things to note is that there is this idea that we are going to use knowledge about haplotypes of individuals to impute information about SNPs that we might not be measuring. So remember, we're only using these 1 million tagged SNPs when we are using these microarray chips and things of that sort. So we're going to have to make some assumptions about all the other things that are associated with this genome and with this haplotype, and the things that are co-inherited with it. To do that, there's been efforts to build specific databases that we can use as references for that. One of the big ones originally back in the mid-2000s was the HapMap project. And there are still lots of other variant curation networks out there. I'm kind of involved a little bit with the ClinGen, the clinical genetic variant annotation and curation network. But you can see, through this type of effort, we were able to identify kind of big differences between various groups. So here we can identify, all the specific SNPs that seem to be associated with different ethnic groups that have very different evolutionary histories, sort of Asians, Western Europeans, and Nigerians. And we can identify all the SNPs that are highly correlated with each of these groups, right? So at a 0.8 or 1%, so these are, basically every individual would have a specific allele, 1.6 million alleles associated with the Yoruba. And as I mentioned before, one of the great things about doing this tagging study is that we can use it to sort of do this imputation, right, where we can make these inferences about these genotypes, which allows us to increase the power of association test. It's specifically powerful because we can just look at a very small number of these sites, and we can basically infer all the intervening sequence. So this is just showing an example of how this might work, right? So we genotype an individual, then we end up with something like this, right, where we have scores, but there's a lot of question marks here, things that are missing. We then can compare it to a reference set of phenotypes, right, or haplotypes. We can figure out what the actual sections are that are associated with the reference, and we can fill in all these question marks. We can go over here and start filling in all the rows that we're missing. So, as we've gotten better at GWAS, we've tried to impute more and more SNPs. And as we've done that, this has required us to have fancier and fancier computers because we've gone from maybe 50,000 SNPs to a million, then to 2.2 million. And in addition to just assessing, to sort of taking imputation as a fact, we've also done a whole lot more work on assessing the quality of imputation. Understanding that, we've got higher rates, higher error rates for imputation than for genotyping SNPs. So it's important to remember that imputation can be kind of an error-prone process, and we need to take that into consideration. And it doesn't really work well for alleles that are rare because since they're rare, they're difficult to observe and difficult to code and difficult to get into consideration. A lot of times it's really important for us to think about the probabilities. Anytime we actually do SNP calling, we should get some sort of scoring or probability structure that would tell us, how good of a call did we actually make. And it's important that we take that into consideration when we think about how well we're able to actually identify what the score is that we have in our data. In the end, you're going to end up with something like this. We've got our log of odds score over here. This is our, this is my log of my p-value here. And so this p-value is indicating, how significant the result was, the association. So that the higher the number, the more significant is the association we observed. And you can see we can go across all these different chromosomes and we can identify specific spots on these chromosomes where there looks to be or a site that has a lot of association with specific outcome. And this is a common way of looking at these results, going along a genome, trying to find regions that are highly associated with some specific type of outcome. So how do we get those p-values? Basically, we're looking for additive effects of alleles. So what we're doing is some type of regression through the different allelic states that could be happening and just drawing a line through those to try to better understand the relationship between alleles and outcomes. When we think about interpreting SNPs, it's important that we think about whether or not that SNP is indicative of any function. Is it a synonymous or non-synonymous change in coding sequence? Is it affecting binding regions for transcription factors? Is there any sort of obvious biological effect that changing that DNA sequence could have on an individual's success? Of course, looking in the literature is always a good idea. Looking at databases like Clinical Variants and ClinVar is also really good. Trying some alternative paths to make sure that that SNP is really significant, not just relying on the association study. There's a lot of things that we can do to sort of better interpret the patterns that we see. And one of the important parts of this process is that we're able to have some level of replication in our data sets. One of the big problems with association studies is that they are hard to replicate. It's hard to go back and do the exact same study that you did before. And because it's not mechanistic, because it's often not controlled, there may not be the same kinds of controls that we might include in true experimental work. There have been a lot of cases where these associations that we've identified have not held up through time. And I think maybe the best examples of these are some of the controversies that have been going on in the psychoanalytical literature, but a lot of classic studies in psychology have not been able to be reproduced or replicated because they are based on associations as opposed to causations, and thus when you change the context through replication can lead to uncertain outcomes. So how have GWAS done? Well, some people would say they've done pretty well, right? So here's an example where a genome-wide scan has identified a whole bunch of different genes that are related to insulin resistance, and so it's been very successful in this case. We look at a meta-analysis of all different types of SNP associations related to blood lipids. We can find, across all these different studies, 59 new associations of SNPs that are explaining 12% of the trait variability and 25% of the heritability. So, having a decent sized effect on this issue. And there's a whole bunch of other cases that we've used GWAS studies and found specific associations that have been really important to us understanding how these diseases work. And yet, there's a lot of problems with them. So, one of the big great hopes of these, as Sir Alex Jeffreys says, is that they were going to pin everything down to the DNA level, right? And they were going to be able to identify all the single gene disorders, and we were going to understand this genotype to phenotype map explicitly. And it turns out that it's just a lot harder than that, right? And one of the problems is that we look at the amount of heritability, of how heritable these traits are, and when we do GWASs, it explains a very small percentage of that data. So where are we? Well, as of the 10th anniversary of the GWAS catalog, there's now been a publicly available curated resource that's been compiled for all human GWAS findings. And in there, there's about 68,000 SNP traits associated from 5,000 studies. So pretty impressive amount of work. And it's targeted a lot of those. Remember I talked about that CDCV proposition. It's targeted a lot of common variants. So common variants, it's been very successful at identifying common variants that are associated with common diseases. However, other stuff, the vast majority of other variants, we don't understand what it does or why it's there. So there's a bunch of reasons or steps that we could take to try to alleviate some of these problems, and that's as we go through and look at the results, what are the effects does this have on, we need to think about the type of approach we're taking and how that might affect our ability to find a relationship between traits and disorders. So is it better to do a case control approach? Is it better to do a cohorted approach, or both? How many samples are we taking? How are we defining the phenotype? This is a big one for things like GWAS is looking at autism, for instance. Autism is a spectrum disorder. It's not just one thing. And when you do research, I think in Australia, you're not even allowed to use the term autism because it's not just one thing. It's not a yes or no. There's so many different ways that it can be defined and it can be expressed. And so trying to coarsely say, I'm going to look for the genes that underlie autism, ignores the fact that the condition itself is not just one thing. So it's really important that we take that into consideration when we're thinking about actually analyzing these data. So if we want to think about some important things that are going to be that are going to affect the comparability of cases and controls, are we using the same methods? Are they genotyped at the same level, same quality? Is there population substructure in cases or controls that are contributing to what we might be looking at? And are all the laboratory procedures actually being consistent throughout the experiment? So as I mentioned before, when we do these case control things, we're going to be comparing allele frequencies where there is disease to allele frequencies where there's controls. And we can see here, we have two different populations. And in these two populations, they look at very different allele frequencies of reds versus blues. So if we actually want to be able to associate this red allele with a specific disease outcome, it's important that we not mix populations because we could really wash out the signal that we're trying to look for. Talked a little bit about this before, about the CDCV proposition, right? So how easy is it to find genetic variations by looking at their risk allele frequency and the strength of the genetic effect or the log of odds ratio. And again, this case where we have common variants that have that are for a common disorder, then they'll be a lot easier to find. Now, the problem is when we move out of sort of this area and we're looking at common variants influencing high effect common variants influencing common disease. There's not a lot of these, but there are a lot of rare variants that have small effects on phenotypes that are hard to associate with genetic outcome. Another thing to take into consideration, right, when we're doing these, we're looking at associations between, maybe 1 million different SNPs at a time, maybe 2.2 million SNPs at one time. That's a lot of different statistical tests, and 5% of them are just liable to be incorrect just based upon chance alone. So we need to make sure that we're correcting for that problem using multiple comparisons tests. A classic one is a Bonferroni test. This is just basically taking your p-value and dividing it by the number of tests that you're going to be doing. This is a very, very, very conservative approach to doing multiple test comparisons. There's a high probability that you're going to have a lot of false negatives in this case. A different approach is an FDR, or a false discovery rate approach, and this is designed to control for the expectation that a proportion of results will be identified incorrectly. And so what this says is that at some value, 1%, 5%, whatever it is, 0.1%, there's going to be a certain number tests that are going to be significant that are really not significant. And what this just says is, well, let's rank these results by our p-values and we'll get rid of the ones that are the largest p-values. So how does this work? We have our p-values over here. We then rank them all by their score, and then we'll say, okay, how many of these are below, if we say below 0.1, after we rank them and correct them, how many of them are still below that threshold. So, there is no sort of magic p-value where there's no error. This is kind of a sliding scale process. Generally, that p-value is going to be somewhere between 10 to the negative 5 and 10 to the negative 9 that we want to make sure that a SNP has. But this can range. It depends on the type of correction you want to use, and it depends on what the purpose of your GWAS is. For clinical outcomes, you want to probably have a very stringent and low p-value. But for something where we're just looking at a heritable trait such as height, we may not need to be so stringent in how we are establishing statistical significance. One of the issues that has confounded scientists when it comes to genome-wide association studies has been the idea of how genetics can actually account for phenotypes. So if we look at something like height in humans, which is about 80% heritable, there were a ton of genome-wide association studies done in the 2000s that identified about 40 loci that can contribute to explaining the genetics of height. And they were all significantly associated with height as a trait. But the problem was that they only explained about 5% of that 80% variation. And this is really problematic. It wasn't just height. There were a whole bunch of other factors too where genetics just was not doing the job in terms of explaining what these outcomes were. So what's going on here? Well, if we think about these, going back to those original equations about how we get a phenotype. So a phenotype is some sort of combination of the effects of genotypes and environments, right? And we think about the variation of a phenotype basically being the result of some variation in genetics and some variation in environment, and then an interaction between those two environments. If we're able to actually say, okay, I'm going to somehow make this covariation between genetics and environment equal to 0, I can calculate something called a broad-sensed heritability. And a broad-sensed heritability is going to basically be the amount of genetic variation divided by the amount of phenotypic variation. Now, we think about genetic variation, there can be a kind of a bunch of different factors that contribute to it. There could be dominance variation, There can be epistatic variation. There can be all these things. What we're interested in this case is the additive genetic variation. So we want to see the effects we want to see be able to isolate the effect of the average genetic effect of alleles on that phenotype. So in this case, what we're really trying to identify is the component of genetic variance which is responsible for parent-offspring resemblance. And what we do is we redefine heritability into something we call the narrow-sense heritability, where we'll only look at the additive genetic variation, or this variation in A. And that's just defined as the additive genetic variation, or the narrow-sense heritability is defined as the additive genetic variation divided by the phenotypic variation. So what does that mean for missing heritability, right? It means that we're trying to look for this component that is contributing to this additive genetic variation, right? That's the main part that we're trying to identify, and we weren't doing a very good job of it. So right off the bat, there were a whole bunch of issues that came to light. So some people said, oh, well, missing heritability, it must be epigenetics, because we're doing such a good job with these genome-wide association studies. It's got to be some sort of epigenetic factor, some sort of inheritable genetic factor that we're not measuring when we do genome-wide association studies. And that could definitely be true. It could be that. But what it turned out to be is it's probably less that and more of an effect of some issues with how we actually conduct genome-wide association studies and where they are appropriate. So, we talked a little bit how there's this kind of sweet spot where it's very effective to use GWAS to identify certain genetic relationships with traits, right? So there are certain conditions where this works well, but there are others where it doesn't, right? And so when we're talking about variants that have small effects, you're going to need really large sample sizes to do that, and that may not be that easy to do. A lot of times we're not finding causal variance, right? We're imputing the role of a specific variation by something that's close to it. So what can happen is we can end up with a difficulty in identifying true positives. Structural invariants are also really important in common disease, and these won't be identified with any sort of SNP-based technology. So arrays, microarrays that we previously were using for this, and even short-read sequencing may not be a good way of encompassing this type of variation. And then there's a lot of other factors that can play a role in identifying disease. There are lots of ways that estimates of heritability can be flawed, either by genome interactions or genome by environment interactions. And all these things can affect our ability to actually explain and detect the genetic component contributing to heritability. So there's a whole suite of methodological issues that sort of came out of this process that were discovered. We talked about some of these earlier, but not taking into proper consideration of population stratification can really bias heritability estimates. Local differences in the genome of linkage equilibrium estimates, the importance of minor allele frequency determinations. So it was found that one of the big corrections that allowed for something like the estimation of heritability attributable to human height had to do with shifting our focus from thinking about identifying alleles of statistical significance that would have sort of a moderate to large allele frequency in a population to these minor allele frequencies. So when we started considering all these minor alleles in populations, we actually did a much better job of explaining genetic patterns of heritability in human height. There's a couple other areas where specific approaches can, like the REML method, which is often used in controlled case data, can downwardly bias some of our estimations of the relationships between phenotypes and genotypes. And then if we don't take into proper account environmental effects, this can also have a big effect on our ability to estimate heritability, as it can make us seem like heritability is a lot larger than it should be, and so thus we're only going to explain a smaller portion of it. But, aside from methodological issues, there's some sort of conceptual issues that also need to be taken into consideration. So, one of the big things for GWAS, especially in the health context, is that are we actually looking at a population and measuring these traits in that population that's going to be able to tell us something about other populations and other groups? So there's been a lot of effort to try to, sample a lot more broadly, especially when it comes to ethnicity and genetic background, and health studies, so that we can better understand. We know that health outcomes can vary dramatically between groups of people, and that may be due to health, but it might be due to environment too. So we can't um, necessarily rule one out over the other, but it's really important that we sample broadly. If we're just looking at one population in a village in Britain, we're not really sure how well that's going to apply to, say, another place somewhere else in the world. It's also important that we keep in mind that GWASs are not necessarily relevant to each individual. They give you population level estimates. And while certainly they can do things like help us identify risk factors, they aren't necessarily fate determinants. They could be, but a lot of times we're not even sure what the causal underlying factors are that are contributing to these traits. And they may just be a co-marker or co-indicator in a specific population of individuals. We need to make sure that we're not separating genetic influence on trait development from the environmental context where we are observing this trait. It's really important that this is the case. So, thinking that we actually are able to keep environment to genotype interactions to zero is probably not a realistic assumption in a lot of cases. And I think that at its heart, though, there's this other idea that it may be a little bit simplistic for us to think that genetic effects can be captured just by looking at single nucleotide polymorphisms. That level of variation is significant to explain the patterns that we are observing. While there certainly are cases where that is the case, where we see the distinct mutations and variations at specific points in the genome driving the genetic control of a phenotypic trait, it is not always the case. Most genes, most traits are not these sort of one-to-one connections. We don't see a lot of these traits determined by alleles of large effect. In fact, we often see traits determined by multigenic, multi-locus effects more frequently. And a lot of times the effect of any one locus might be really small. And so it's these polygenes and these interactions with environments and this more complicated view of actually how genetics works in the context of the world, which is really important. GWASs are great tools for identifying and surveying relatively quickly, a population and a sample for a relationship. But it's just an indicator of what that relationship might be. It's really important that we take that information and dig in more deeply to try to better understand what's driving this process, what's driving the trait that we are interested in observing, whether it be a disease or some other sort of phenotypic trait that we're interested in.